Okay, hi, my name is Minji.
My name is Catherine. Sorry.
My name's Julia.
And we're going to talk about a topic that we covered on Wednesday, which was on biotoxicity. So for this lab, we were using silver and yeast particles to see the effect of silver on the yeast cell. And to do this, so we used 3 particles to do this. And the first one was silver powder, and the second one was silver ions, and the third one was silver nanoparticles, which were most effective.
Okay, so for our drawing, there are 3 main ideas. And so for the boulder, that represents the silver powder, and the wall represents the yeast cell. And so what happens is that the silver particles are trying to infiltrate the wall, right, which represents the cell. So since the boulder is too big, it won't go through. And the 2 smiling people on the left side over there, those represent the organelles, and they're happy because they're able to defend the yeast cell and prevent the boulder from infiltrating. And the 7 bars on the top left, that represents the health of the cell. And so since there's a full 7, that shows that the cell is pretty healthy and there's no damage done. And the middle section, that represents the silver cations. And since they're ions, they have to pass through the ion channel. And since they have to go through an extra step, the person is drawn shooting them from a slingshot. And these ions are able to go through, but only some of them are effective, which shows that some are past the wall and some aren't. And the health of the cell is decreasing, so it shows that the cell is getting damaged. And the organelles are a bit freaking out because there's damage done. And the last section shows the nanoparticles. And the giant one shows how there's a bunch of nanoparticles making up the ball. And these kind of show how, these nanoparticles kind of show how they're passing through the wall and they're able to make it through, and since they're disguised as the glucose molecules. And since they're able to infiltrate the wall, they splatter on contact with the cell membrane, which makes the nanoparticles destroy the cell from within. And so this effect is displayed by the 2 organelles being with X's in their eyes, which shows that they're dead. And yeah, so it's a bit morbid, but that's what happens. And then,
Okay.
yeah, and then also the health of the cell is decreasing, which is represented by the 2 bars. Yeah.
Okay, so basically how the silver is able to have this effect on the cell is because silver is used to prevent and protect against like bacterial growth and stuff like that. And the process of how they kind of do that is like basically all these different types of structures, like the powder, the ions, and the nanoparticles. So they all eventually are basically they break down into like kind of how the ions are. And because these ions have a very high amount of like surface area, they're able to do the most damage. And so like the boulder in the example, so that represents the silver powder, and that will eventually break down into those silver ions. So it's eventually able to do damage, but but when it's only powder, it's not able to really be effective. So, and then the silver ions, so those are able to do damage by just like mimicking kind of like the cations that enter the ion channels in the cell's membrane. And they're able to do some damage. But the nanoparticles, so what they do is when they make contact with the cell wall, they're able to release the smaller silver ions, which are able to do damage to that, because the silver ions, so they catalyze the formation of reactive oxygen species, And what the reactive oxygen species do is they are able to react with the lipids, causing the membrane to break. So that's how the nanoparticles are able to infiltrate the wall. And so additionally, the display doesn't necessarily show exactly how the particles are able to actually like get through to the cell wall, because when you're talking about cells, like for example, like the nanoparticle. So the silver nanoparticles, because they're like a very similar shape to how glucose molecules are, the cell actually takes them in through endocytosis. So in a sense, it's a lot more effective than these other ways that are shown, which I don't think was exactly like explained as well through the drawing. And also, in addition to that too, we learned more about the surface plasmon resonance, which is basically like when the molecules are smaller, the electrons, they move faster, which makes a higher frequency for the wavelength of light. And when they're bigger, the electrons make like a slower frequency, which means that the light will have like a lower frequency of like the wavelength will have a lower frequency. So that means that like the bigger the particle, like it's more close to red light versus a smaller particle, which will emit like a color closer to blue or violet. So, I think we're open for questions now.
Are there any questions?
Yes.
So the silver is trying to go through the glucose or yeast cell because they're trying to destroy the yeast cell from within. So even though they go in through the membrane, they still kind of destroy the organelle and basically kill the yeast cell overall.
Yeah, they still have a toxic effect on what's inside the cell.
The middle section right there.
Oh, the bowl, right?
Out of the way.
Now it all makes sense.
Yeah.
Now we get it.
Yeah.
Okay, so you guys said, so in agreement with what I said on the first day when we talked about cartoons, they can never capture everything about experiments. And so you described some things that were not captured, in particular the idea of the nanoparticles being eaten. So how could you possibly imagine modifying this cartoon to add some aspect of the experiment that was not already shown?
Maybe there could be like a gate or something that passes through, like to get like supplies or something like that, and maybe that could be like a way for, like, for example, like for like the ion channels, we could do something like that. So it's kind of more obvious that it's through like it goes through something in order to get through to the cell, not just breaking through necessarily.
And in addition to that, maybe this person could have added an additional wall or something that represents the cell and the glucose entering in to show a comparison between the nanoparticles and the glucose.
And also, like, since the nanoparticles are kind of disguising themselves as glucose molecules, it seems kind of like the nanoparticles are just like a rock. It doesn't really act like the wall itself.
Oh, like I guess the special thing about like the size of the nanoparticles that we're using is that they're like the same size as the glucose is. So it's actually, I believe it's more effective in that sense because it's kind of, it takes on a similar like shape and size to what like the cell wants, like the glucose that it wants. So.
It's a cluster of nanoparticles.
So we actually heated a solution of silver ions, and as we heat the silver ions, they cluster into small particles. And the instructors told us that if we get like a slight yellow color, that indicates that we have small nanoparticles, like Julia said before about this plasma effect.
Yeah, the plasma effect, the surface.
Yes.
Well, so because like the size of the particles like determines kind of like the wavelength frequency, so we could like, are you asking why, as in why yellow would be a good color, or as in just? Oh.
Because as the color gets darker, that means the nanoparticles are getting bigger. So since it's yellow, it means that the nanoparticles are still relatively small.
Yeah, it means that.
What's the actual size scale relative to each other, like from your largest molecule down to your nanoparticles?
So the silver powder would be the biggest, and then it will be the nanoparticles, then it will be the ions because the nanoparticles are actually composed of tiny silver ions. The nanoparticle would be around 10 nanometers, which is similar in size to the glucose. We're not sure about the other two.